I will talk about neuroplasticity in normal brain physiology and disease, and this title also can seem rather broad, but you will see that I will focus to what is known today about roles of transmembrane cell adhesion glycoprotein neuroplasticity in some of very important processes for the functions of human brain and mammalian brain, and this is neuroplasticity, neuroendocrine homeostasis, neurodevelopment, and neurodegeneration. So first we will start with questions, what do we know about neuroplasticity? And probably the best thing would be just to give some information about gene annotation and protein isoforms annotations. So what we know, of course, is at which chromosome the gene for neuroplasticity is situated in different species. I put here the information about mouse, rat, and humans. And what is important also to note here is that we have two isoforms which are the products of that single gene. And those two isoforms are differentially expressed in different kinds of tissues. And you see also different names for those isoforms. These ones which are in bold letters are the ones which we utilize the most. And I will now tell you what's the main difference between those two isoforms. If we talk about neuroplasticity, then first we have to repeat that we are talking about transmembrane protein, which is highly glycosylated, and that it is a member of the immunoglobulin superfamily of cell adhesion molecules. I don't know whether I have some pointer here. Okay. Yeah. So this is the one. Okay. Very small red dot, but I think it will do the job. So what is the difference between those two isoforms is that if you see it, the isoform 65 has 3 Ig modules, and they can be expressed in the neighboring cell membranes, so they can make some interactions between two membranes. While on the other hand, this smaller isoform, which is NP55, can make so-called just cis interactions with the partners which are at the same membrane. So those two isoforms basically are differentially distributed, and this is interesting because they can have then different specific roles. Brain-specific neuroplasticity 65 is really expressed only in the brain tissue, and the other form, which is the smaller one, is ubiquitously expressed, meaning it can be expressed in many other tissues. Anyway, the name itself, neuroplasticity, has been annotated when first studies show that we are talking about glycoprotein, which is highly abundant in the synaptic membranes and postsynaptic densities. And then the whole series of studies started, which evidenced and strongly supported the idea that the cell adhesion glycoprotein molecule is important for many important processes in the brain, like neurite outgrowth, synaptic plasticity, maintenance of long-term potentiation, etc. So when we are looking at where exactly this protein is expressed, then we can see it is expressed in presynaptic and postsynaptic membranes. So this is the nice picture which is related to studies of hippocampal excitatory synapses coming from the red hippocampal neurons. And also what is interesting as the information about neuroplasticity is that it makes several binding interactions with different membrane partners. Some of those partners are FGF receptor, we talked also about this one, GABA A receptors, and monocarboxylate transporters, which is quite important for the energy metabolism of brain cells. And one of the more recent studies and papers show that there is also one additional binding partner for neuroplasticity, which is plasma membrane calcium ATPase. We will talk it, of course, as PMCA, it is easier to say. This interaction is at the structural level, meaning that neuroplasticity has been shown to be the obligatory subunit of the PMCA, meaning if the neuroplasticity is not bound specifically within the membrane part of this PMCA, then the PMCA doesn't work properly. There are, of course, several suggested signaling pathways which are activated by neuroplastins. And what has been shown firstly is that the signaling cascade is dependent on the interaction of the FGF receptor with neuroplastin. And you can see some of the generally activated downstream kinases, which are basically shared by many other important cellular processes. So some of those kinases are also activated by the signaling cascade of the neuroplastin. And some of the more newer data, more recent data, show that we have interactions with some other molecules like TRAF6, which is important for the spinogenesis, meaning very early in the development of the brain cells, when dendritic spines are being formed, then this interaction of neuroplastin with TRAF6 is existing, and when we have adult neurons, then this interaction is not anymore present. And also there is another interaction which is important for the people who are doing with inflammatory or neuroinflammation field or inflammation immune response in general, is that interaction of neuroplastin and PMCA stabilizes PMCA, increases its catalytic activity, and regulates T-cell calcium homeostasis following T-cell activation. So this was just some information about the signaling mechanisms which are activated by neuroplastin. Also, what is interesting to say about neuroplastin is where this protein is expressed when we are looking in brain tissue. So data are coming from studies on the rodent brains in mouse, rats, but I will show you something which is coming from the studies in mouse. What is known is about cellular and regional distribution, and what is also very important to note here, that when we are talking about cellular expression, then neuroplastin is expressed only on the neuronal membranes, so not other types of cells, like glial cells, they do not have this NP65, just neuronal cells. And when we are talking about regional distribution of expression, then although there is a widespread expression in the mammalian brain, it seems that the highest abundance is in the hippocampus and cerebellum. What is very important to know is what would be if neuroplastin is not expressed, of course, in the brain tissue. And there is a group in Germany who developed the constitutive and also induced deficiency of neuroplastin in mice. So this mouse model gave very important information of why neuroplastin is crucial for normal functioning of the brain. So what they showed is that neuroplastin is involved in learning and memory, and also in hormonal homeostasis. You have here the list of the main phenotypic characteristics of neuroplastin deficient mice. I will not now read them all, but for instance, they have reduced lifespan, corticosterone elevation, and what is mostly important is that they have impaired associative learning and memory, meaning that they just cannot retrieve some learned associated memory. So some of the quantitative data coming from those behavioral studies would just be in support of the result that they have reduced context memory. Also, when you look at what is the social interaction of those mice, so whether they want to have interaction with another mice, to move to another chamber, what has been shown is that somehow they just don't want to mingle, okay? So the social interactions are not really good in those neuroplastin deficient mice. And what is related to this hypothalamic pituitary axis and the neuroendocrine homeostasis, what has been shown is that in the morning, when you measure the serum concentration of corticosterone, then in mice which do not express neuroplastin, they have really elevated morning serum concentrations. And also when you make dexamethasone challenge test, then again in those animals you have elevated, increased concentrations of corticosterone. So this was about mice, and what is interesting now for us is what is going on in humans. And rather for a long time we did not have information about neuroplastin in human, just in several studies which were talking about genetic polymorphisms in humans. And what is interesting is that they found some kind of relationship or association with intellectual ability in adolescence, and also in situation when we can talk about susceptibilities to schizophrenia. But okay, there have been some data about genetic polymorphisms, but not where exactly neuroplasticity is situated in the brain. So then we made this expression analysis by just using classical immunohistochemical methods, and we focused to the hippocampal tissue, of course, because this is the site of the processes of learning and memory. And what we have shown is that neuroplasticity immunoreactivity in adult human hippocampus is associated with all those layers which are important for the excitatory glutamatergic pathway, which we call trisynaptic pathway. If you just go into more details, into higher magnification, then we can see which type of cells express and in which layers of the hippocampus. So all there we see this high abundance of the neuroplasticity expression. Also, if you just want to see at the level of the Western blot technique, what is the abundance of this neuroplasticity in differently prepared fractions of the membranes, and you compare it to the total homogenates of the brain tissue, you see that all those preparations in which we have like synaptic membrane, synaptic junction, postsynaptic density, that there is a high signal in neuroplasticity. So all those fractions are enriched mostly in neuroplasticity. Of course, definitely it would be interesting to see what is going on during development of the human brain with neuroplasticity expression. We didn't have enough of the sample for making this kind of analysis, so this is preliminary and unpublished results. But what is shown is that we have some kind of the temporal pattern of the expression of neuroplasticity during different phases of the development of the brain, and it seems that the starting point coincides or roughly coincides with the formation of synapses in the human brain. But what we could do is this kind of spatiotemporal transcriptomics analysis, just using or extrapolating the microarray database of healthy human brain, and then you can make these nice heat maps which show you the expression of selected genes in different periods of the development, in different regions of the brain, and we did that analysis for neuroplasticity gene and for the genes for the 4 isoforms of the PMCA. And what you can see is it is quite evident that the expression of neuroplasticity is rather high in all those different developmental stages in the human brain. Also, what we were able to do is some kind of the comparison between 3 selected regions, neurocortex, hippocampus, and cerebellum, in which we show also this high expression of the neuroplasticity. So this blue line above is the neuroplasticity expressed really so highly as some of the housekeeping genes, in fact. Okay, so later on then we wanted to see what is going on in neurodegeneration. We did so also the changes in the expression of neuroplasticity in the hippocampus affected by the Alzheimer's disease. What we did see is that this expression is related also to the phase of disease. Basically, there is increased expression of neuroplasticity in the early phase of the disease, which we discussed or suggested to be some kind of the compensatory mechanism during the early stages of the disease. But later on, when you have the, the high atrophy and the loss of the neurons and a lot of plaques and a lot of tangles, then of course we have this negative correlation of neuroplasticity immunoreactivity with the number of those plaques and tangles in the human hippocampus. So another aspect of our research is also related to how important is the lipid environment in the membranes for the function of the transmembrane proteins. This is something which is known, of course, from the literature, that they are partnering, they are interacting within the membrane, and that if you change somehow the lipid milieu in the membrane, this will influence on the positioning and on the function of those transmembrane proteins. And we did really see that for the specific class of the lipids in the membrane, which are important for the positioning and evidently for the functions of neuroplasticity. So in order to see really is it true, or how much the gangliosides or those lipid classes could be interesting or important for the functional assembly between PMCA and neuroplastin? We made firstly those co-localization studies. Okay, so there are different species of gangliosides. I will not bother you with that. We found one of those species which really nicely co-localizes with neuroplastin. So we can say, okay, GM1 is important for neuroplastin and then also for the PMCA because there is a functional assembly. But how we really evidence that is that we measured how much of the calcium really, or how quick the calcium is exposed from the cell, because the PMCA is involved in regulation of the cellular calcium concentration, and if PMCA doesn't work properly, then this calcium just don't go out as it should. And this really happened when we blocked GM1. So if GM1 does not have physiological functions, if it is not interacting with neuroplastin and PMCA, then PMCA doesn't work properly. So in fact, very close to the end of the lecture. So the findings which I showed you were related, of course, mostly to the brain tissue, but this is just to know that there is another isoform which is expressed in other tissues, and quite interestingly, this isoform NP55 is important for malignancies, for different malignancies. So basically, main conclusions would be that neuroplastin is important for making those assemblies and functions together with the PMCA, which controls how much of the calcium is inside the cell. So it is some kind of the housekeeper of neuroplasticity, it is changing neurodegeneration, and it is probably involved in neuroendocrine processes. There are several avenues which can be proposed for some future work, and there are a lot of them, so I will not go now into details. We can discuss them later. And the last slide would be, okay, this nice photo of the front of the Croatian Institute for Brain Research where I work and where the majority of that work has been done. This is a small group financed by our Croatian Science Foundation within the project which we called NeuroReact, and also there is a list of our friends and colleagues from other institutions to whom I'm really thankful for collaboration. Thank you for the attention, and I'm ready for your questions.